When all the supplies and reagents are available, the equipment is available, then the next step is to proceed further to determine the quantity of DNA. As a general rule, अगर absorbance value, जिसको हम आम फहम में OD value भी कहते हैं, अगर ये OD value 1 है, इसका मतलब है कि DNA sample की जो concentration है 50 micrograms per milliliter है. इसके साथ साथ क्वांटिटी के साथ साथ हमें जो चीज़ देखनी है या जिसका ख्याल रखना है, हाउ प्योर इज़ द सैंपल. इसमें किसी किस्म की इम्प्योरिटी, किसी किस्म की कंटेमिनेशन तो नहीं है. कंटेमिनेशन और इम्प्योरिटी कैन बी डिटरमिन्ड बाय ऑब्जर्विंग OD रेशो और एब्जॉर्बेंस रेशो at 260 wavelength and 280 wavelength. For a pure double-stranded DNA sample, this ratio is 1.8. If the ratio goes up or if the ratio goes below 1.8, that indicates that there is contamination. Any value of this ratio less than 1.8 reflects the presence of phenol or presence of protein in the sample. If this is the situation, then we have to repeat the process of purification of DNA. And if the contamination is of RNA, the value of the ratio is going to be more than 1.8. So it is advised that you should check the absorbance ratio before proceeding further. If this is 1.8, proceed further to determine the quantity of DNA. For determination of quantity, सबसे पहले हमें स्टैंडर्ड DNA सैंपल यूज़ करते हुए एक स्टैंडर्ड कर्व बनाना है. स्टैंडर्ड कर्व बनाने के बाद वो स्टैंडर्ड कर्व हम फ्यूचर में यूज़ कर सकते हैं अननोन सैंपल्स की कंसंट्रेशन, DNA कंसंट्रेशन डिटरमिन करने के लिए. एंड फॉर दिस पर्पस द एब्जॉर्बेंस शुड बी noted or the optical density should be observed at 260 nanometers. And now, once we have decided that to proceed further, we should dilute the DNA samples using TE buffer, and the dilution ratio is 1 to 100. This graph shows you that how you can read the standard curve of DNA to determine the quantity of DNA in unknown sample. This red line is the optical density or absorbance which is taken for an unknown sample from the spectrophotometer. Draw a line from here and look where it crosses the standard curve, and then draw a vertical line to read the quantity of DNA per milliliter or per microliter of the DNA sample. Using this standard curve, now determine the purity and calculate the concentration of the DNA in any unknown sample. And this concentration now can be used for further experiments which you want to do using this DNA sample.